human cognition, and I should say language here because I decided to focus on language for today with neurostimulation. And please feel free to interrupt whenever you think there's something that needs to be clarified or if you want to say something. So our group is mainly interested in flexible network recruitment during language processing. So we first need to define what we mean by this. What's language in our brain? So this is data from a meta-analysis that we've been doing for over, 8 years or so, where we summarized all current fMRI studies on language representations in our brain. And the very simple and obvious take-home message is that language is organized in distributed networks in our brain. So we have the left hemisphere, we have contributions from subcortical regions, right hemispheric regions, and the cerebellum, which is often neglected. Now, the key point is that we have specialized subnetworks which contribute to different aspects of language or different linguistic aspects, such as the processing of semantics, the meaning of words and sentences, or the processing of phonology, the sound of words. Now, successful communication, when we talk to each other, when we listen to each other, requires flexible interaction between these specialized subnetworks. And now the big question of our group is how the language network adapts to challenges. By the way, I think I should use my pointer so that you can see where I'm pointing. So what do I mean by this? What I ultimately want to do, and where I think I share passion also with Stephanie, is to unravel flexible adaptation to challenges in the language system across different brain states. So in healthy young brains, challenges can naturally arise if you're, for example, in noisy listening conditions and you can mimic this with speech and noise paradigm. We can also artificially induce challenges with neurostimulation. So we use inhibitory, non-invasive brain stimulation and see how the brain adapts to this. And why is this interesting? Because with this, we can build models that we can then transfer to other brain states. So we can mimic how, for example, the brain may adapt to brain lesions or how aging brains may cope with some challenges. In aging brains, we are interested in dissecting those patterns that reflect compensatory reorganization, because we know that as we age, we have some cognitive decline and the brain has to adapt to that. And we want to dissect such patterns from the decline itself, so from the so-called de-differentiation of brain networks. And finally, in lesion brains, we are interested in which parts of reorganization that we observe help to recover function. So for example, after stroke, which parts of the reorganization of the networks is associated with good recovery. We're also studying this in brain tumors now. So the overarching goal is to uncover common mechanisms of flexible adaptation across the adult lifespan, from healthy young to aging to lesion brains. And now the question is, how can one do this? And here neurostimulation comes into play because it's a simple or relatively simple method where you can perturb certain parts of your network of interest and then look at relatively rapid processes of adaptation. And when you look at classical neurostimulation studies in the literature, then it often comes with the promise of causal inference. So the standard ratio is that I perturb a certain key region, for example, a language region, with non-invasive brain stimulation, here illustrated for transcranial magnetic stimulation. And then I look at stimulation-induced changes in task performance. And the standard ratio is that if the area that I target is really necessary for the task that I'm interested in, then the task performance should be significantly affected if I disrupt this processing. And this would allow for the conclusion that the targeted area is causally relevant for my task. So the naive ratio of many non-invasive brain stimulation studies, be it TMS or transcranial electrical current stimulation with TDCS or TACS, is that causality can be inferred if inhibitory neurostimulation over an area of interest significantly affects task performance in my task of interest, and hopefully this is specific and does not affect a control task, because otherwise you could also hit the brain with a hammer and then you also get disruption. So this is what most studies infer, or what the ratio of a lot of studies, especially in the early decades of TMS in the study of cognition, is. Reality is, unfortunately, as usual, a bit more complex. So here you can see a sketch of a more realistic chain of causation for neurostimulation studies. So it all starts with your non-invasive brain stimulation protocol that could be any NIPS protocol that induces or produces an electrical field in the underlying brain tissue that you see here. So you induce current in your brain. Now, the electrical field evokes or modulates the local neuronal activity in your target region. This means you're not targeting single neurons, of course, but hundreds, thousands of neurons. Now, the activity from that you induce spreads via synaptic connections to neighboring areas of the connected network. And unfortunately, not only to the network of interest, but it can also affect other areas that you're not interested in. And then finally, the activity affects the task relevant cognitive processes that you're currently measuring. For example, if you apply TMS during a language task, then it hopefully affects the language task, and which eventually then results in a response as part of the task. So what I want to illustrate with this, from this simple perturbation directly affecting the output, this is much too simplified. There is a long chain of processes that are in between. And at each step in this chain of causation, we can also have unwanted effects in other parts of the network. For example, your electrical field that you induce may not be as focal as you think. The modulated task-related and network-related activity may also spread to other parts of the network. Plus, we also often have unwanted side effects of the stimulation. If you stimulate muscles or nerve, you can have unpleasant side effects that draw attention away. You have a clicking sound. And for all these effects, you need to control, otherwise they will crucially affect your behavioral outcome. Now, this is to illustrate that the effect is not only influenced by a very complex chain of causation, but there are also influence factors at multiple levels that are often ignored. For example, the physiological parameters of the brain will impact and interact with your stimulation effects. For example, the individual brain morphology, the gyrification, which can vary across subjects, will strongly impact your outcome. And you can model this, but it is often ignored. Then you have methodological parameters. These are usually easier to control. You can set your stimulator intensity. You can choose the parameters that you want to use for your stimuli, but often their impact on the outcome is also not well explored yet. It's sometimes just you choose what others also have been done. And finally, the cognitive processes themselves will also interact with your outcome. For example, the cognitive state. It makes a strong difference if you stimulate subjects while they are at rest versus when they are in a task state. Then also the individual baseline level of performance. It can make a difference if you have good performers versus bad performers. So the take-home message here is that the neurostimulation effects will strongly depend on numerous internal and also external variables. And some are hard to control, and the whole parameter space is not explored yet. And the interactions of these internal and external factors are often ignored, which can lead to unexpected effects, paradoxical effects, or sometimes just null effects. So after this frustrating introduction, what can we do to make things better? I think one way is to use multimodal studies where we're not only relying on behavioral measures, but have multiple outcome measures to better get an idea what the neurostimulation is doing and also how it is affecting the network of interest. And one way of doing this is to use neuroimaging or also electrophysiology, such as EEG, as readout to map our stimulation after effects. And the classical way of doing so is as follows. So you have your, for example, your inhibitory TMS protocol again, and we can use protocols where we induce plasticity. That is, the stimulation effect will outlast the duration of the stimulation for, let's say, roughly 30 to 45 minutes. And then you can map the after effects at the network level. And this is often referred to as short-term reorganization or adaptive plasticity. That is the response of the network to my perturbation. How does the network adapt to the perturbation? And an advantage of this method is that you get an outcome measure beyond the simple behavioral modulatory effect, which is sometimes super small. And finally, we can also combine neurostimulation and neuroimaging or EEG in a simultaneous fashion. This allows to quantify the immediate responses at the network level. And it's, for example, interesting if you want to map changes across the time course of a learning experiment and are interested in differences at different time points, then you can see how, if you apply neurostimulation directly during fMRI, how this changes the activation across the time course of your experiment. So both combinations quantify the impact of neurostimulation at the neural network level, and we can look at changes in task-related activity, for example, during a language task-related interactions or effective connectivity, and associate them with behavioral changes. You could, for example, of course, also include resting state and quantify the impact of neurostimulation during rest, and then try to predict how this will individually affect task performance. So this is the basis that we need to understand how we can use such approaches in the language network. And several studies have mapped such short-term plasticity, so applied TMS first, and then looked at induced changes at the network level during different language tasks. This is a summary of some studies that have in common that they targeted a single key area for language, or one study also auditory cognition, with inhibitory neurostimulation, and afterwards put the subjects to the MR scanner and looked at induced changes in task-related activity. And what these studies have in common is that they show that after perturbation of a key region, such as the IFG, they found an upregulation, so stronger activity in homologous areas. And such changes in the task-related activity are usually taken to reflect compensatory activity increases after disruption of the dominant, and in case of language, mainly in the left hemisphere. So the idea is that the brain flexibly adapts to the perturbation, to the inhibition of one hemisphere by upregulating the other hemisphere. So based on these studies, I have recently suggested a model for flexible adaptation in the language network. And this model comes with two key assumptions. The first key assumption is that flexible adaptation can take place within specialized networks. So let's assume we have a simplified language network with several regions, and we target one of these areas with inhibitory TMS. This is sometimes referred to as virtual lesion because you perturb the ongoing activity. Then this will decrease the contribution of the targeted area. And now the brain may adapt to this. And this looks as follows. We sometimes see, after perturbation of one area, an upregulation of the remaining network regions, which would point towards a certain robustness of the network. So the other network nodes may simply work harder to still be able to perform the job. We also sometimes see a shift of the functional weight towards other areas of the network. This may be areas that have contributed more subthreshold before and are now unmasked in their contribution. And finally, as illustrated before, we also often see an upregulation of homologous areas. And this upregulation of homologous areas is still widely discussed in the literature. Some researchers think it's adaptive, others think it's more maladaptive because the non-dominant hemisphere may be released from the transcallosal inhibition of the dominant hemisphere. And I think here relating such changes to behavioral modulations help to understand what's going on. So the second mechanism that my model assumes is flexible adaptation across different networks. Here, the idea is that sometimes when we target key areas, we don't see focal inhibition of the targeted area, but rather widespread inhibition of the larger network. So if the whole network is decreased in its contribution, then of course you can't see flexible adaptation within this network because it's downregulated. And in such cases, we sometimes observe the recruitment of alternative networks. This could be different pathways, for example, during reading when you have a dorsal or a ventral route, or when we inhibited the semantic network, we saw stronger contributions of phonological working memory pathways. And finally, and this is something that is most interesting to me at the moment, we also see that after perturbation of the specialized language network, a stronger recruitment of domain general networks. So domain general functions such as attention and especially cognitive control and working memory are known to contribute to all higher level cognitive processes, including language. And the idea is that disruption of the specialized network may to some degree be compensated for by stronger recruitment of domain general resources. I'm of course not suggesting that domain general networks can replace specialized networks, which is biologically implausible. Now, two questions that remain unsolved so far and that I'm very interested in is if such flexible adaptation is a general mechanism that occurs across different brain states. So do we see similar mechanisms that we here infer from non-invasive brain stimulation also under other challenging conditions, such as the aging brain and brain lesions? And finally, and most importantly, what is the behavioral relevance of such compensatory changes? Is it just a shift in the balance that we nicely met with fMRI that has no consequences, or is it relevant? And I already see a first question, which is great. So please go ahead.
Yeah, thanks so far. I could very much follow. I just had a clarification question concerning the method kind of behind measuring these networks. Because if I understood correctly, it's mostly EEG you use after TMS application, right?
This is all fMRI work.
fMRI, okay.
Yeah.
But so I was just wondering, because those networks might be really close to one another, how you can disentangle on an individual level, which node now. So if it's actually down up regulation within a network, or maybe within another network. So how focal is it that what you're measuring?
Exactly. So this is a good question. So we mainly use fMRI, where we have a relatively good spatial resolution, and we also use predefined network templates or parcellations to check in which network configuration we are. So because it makes it different if you can call something within network or between network compensation. Yeah, that's a good point. This may be more tricky for EEG data. We haven't done much in this direction with EEG data yet. We are on the way to do so. And there the idea would be to combine this with information from fMRI data, so to also have the network outline. For EEG, of course, it's one pro argument is that we can look more at the time distribution and the time dynamics, which are super interesting, which we completely ignore here. Yeah, this is important. Yes,
Thanks.
Another question.
Yeah, I forgot that I can ask straight away. So
sure.
So the previous slide when you talked about homologous region, so did I understand correctly that when you are inhibiting, for example, left hemisphere, then you have increase of what? Bold signal, right?
Exactly. Yeah. And the studies that I used to inform my model showed increases in the bold signal in the right hemisphere, so in the homologous areas, but we also saw increases in connectivity, so in the influence from the homologous region on the targeted region. So we saw in one study that if you disrupt the left IFG prior to a language task, then you get stronger upregulation of the homologous right IFG, and the right IFG also increased its influence on the left IFG, and this increase in the influence was facilitatory. So we modeled this with dynamic causal modeling. And now you may ask, what good is it if the other area increases its influence on the perturbed regions because it cannot perform the job anymore. But here it's important to keep in mind that when we perturb something with non-invasive brain stimulation, it does not mean that the area is silenced. So we shift the balance, we inhibit this area, and we can also measure this as less activity, but it does not mean a lesion or complete null activation. So it's more that the area has relative less contribution. So that's why I would always think that this is a change in the dynamics of the network. Does this answer your question?
Yes, just a lot of thoughts. Yeah, I need to think. Thank you.
Okay, so after we have the basis and the model of what we need, now it's getting a bit more complex because I want to elucidate flexible neural network recruitment in aging brains and the role of domain general networks here. So, and before it makes sense to apply neurostimulation here, we first need to find out what the default state in the aging brain is, or which of the changes that we observe may be good, reflecting compensation, versus bad, maybe reflecting the general cognitive decline or the de-differentiation of the network. So what do we know here? Here I have an example study from Sandra Martin, who is a postdoc in my group now, and who was interested in neural reorganization in the aging language network. So she used a verbal fluency task. So she had semantic fluency, so a task that has relatively high executive control demands. Some people would even call it an executive task rather than a speech task, but that doesn't matter for now. So what she found was that when older adults perform semantic fluency during fMRI, we see increased interactions between domain general networks and domain general areas, which is associated with better task performance. So what did she find? This is data from task-related activity, so fMRI activity, where we see that old subjects have more activity during verbal fluency. So this was an overt-paced fluency design during fMRI, and we just measured task-related changes. And we see that older adults show increased task-related activity in domain general areas. So also here the right IFG, and some of these midline structures and SPL. Older subjects show also task-related interactions between domain general networks during this task. So this is data from a psychophysiological interaction. So we modeled the interactions of different network nodes during the task that were active. And we see that during the task, we find increases between nodes from the so-called multiple demand network, so the cognitive control network, and the default mode network. And we also have reasons to believe that some of these changes are associated with good performance, because we found that increased interactions inside the domain general multiple demand network, so this network for cognitive control, is associated with better executive functions that we measured outside the scanner selectively in the aging brain. It doesn't seem to be significant in the young brain. So these data give some first hints that some of these changes and some of these increases in domain general network interactions are associated with good behavioral performance. But this is just general executive functions. What about as fluency itself? So to address this, we reanalyzed the data with a network approach. And we also found that increased facilitatory network interactions predict better verbal fluency in older adults. So here we investigated the same data again. So we look at task-related activity during semantic fluency, and then used an independent component analysis to extract the underlying networks, and then correlate the time series between the identified networks. And what looks a bit complicated has a relatively simple take-home message. Namely, we see age-related differences in network interactions, such that we have increased facilitatory network interactions between networks that are usually anti-correlated. What does that mean? If you look at these older adult plots, you see that all the interactions are positive, so they are positively correlated. And these are also interactions which are usually anti-correlated, so negatively correlated in young brains, such as interactions between default mode subnetworks and ventral attention networks, so networks that are usually anti-correlated. And these changes seem to be associated with better performance. So facilitatory network interactions predict or are associated with better semantic fluency selectively in old brains, whereas this is different in young brains. So these changes seem to be adaptive or of behavioral relevance, at least when it comes to the task accuracy. So this gives some hints that some of these changes that we see as we age, especially more interactions between networks that usually don't go together or which are usually anti-correlated, are recruited or associated with better task performance, at least with a task that has high executive demands. And this may indicate that older subjects use different strategies. They rely more on the networks that are less needed for good task performance in young subjects. And this is data from a graph theory approach where we could back this up with other measures, which shows that older adults also show less segregation between networks. What does that mean? Here you can see the visualization of the graphs for the different networks in young adults and old adults. And you can then quantify a measure which reflects the segregation. That is, how much networks form specialized communities. And you can see that older adults have significantly reduced segregation between networks. That is, they have more interaction between the networks and less specialized communities, so to say. So, and finally, when it comes to the behavioral relevance or the correlation with good performance, we see that a high degree of segregation or specialization of the networks is associated with better language accuracy and better executive functions. So both with semantically demanding performance and general executive functions, selectively in young brains. So it seems that a high degree of segregation is good for young brains, but potentially not so good for old brains. And I see a question. Please go ahead, Valentina.
Yes. So these results are related to language, not a language task, but it's actually, I mean, they could be generalized to a broader network of cognitive functions. I mean, we are seeing all these involvement of other networks, such as ventral attention, dorsal attention. So I guess it could be true also for other tasks. It's really a general reorganization of cognition.
Yeah, that is very likely, but of course we only tested it for semantic fluency here, and we chose this task deliberately. So you might also say, yeah, but semantic fluency, it is an executive task. So what you're showing is only an executive component. But I think it is interesting that even I mean, still the semantic fluency task requires that you get the members from the category. So you see, I don't know, birds and have to come up with birds. So it has a semantic component, but I agree that it's very likely that it is a very general function that we see here. But to make claims about this, I think we should include other tasks and see if we check if we see similar mechanisms.
Yeah, exactly. Both the nature of the task that you are measuring and also in general how the brain works, right? We have all the contribution of other cognitive functions to
exactly. So it could be that it is. So what would be interesting, for example, if we see similar patterns when we have phonemic fluency, where you don't rely on the semantic categories, but we didn't test that. So I would think some of these mechanisms are highly transferable and just, as you said, reflect the general ability of our brain. So how much language specific this is, I can't say yet, or not with this data, I should say. Yeah. Stephanie?
Yeah, so I may have missed it, but you separate them into older and young adults. And I was wondering if you know what the cutoff is
I think the cutoff
when
was
does brain change.
That you mean.
Words.
No. Yeah, that is a super interesting and cool question. So what we did here was young subjects are the typical students, so 18 to 35, and old subjects are older than 65 to, I think we had 65 to 80 here, if I remember correctly. And then what would be super interesting is this middle-aged subjects, right? So this is something that I'm very interested in. I know that Jonas Obleser has done a lot of work with these middle-aged subjects when it comes to their hearing loss. So he always uses these speech and noise paradigms and then checks how much the semantic content of sentences helps, or how this interacts with the performance under degraded listening conditions. And there, I think he has some ideas that this that there may be a gradual change rather than a direct cut. If you cross this line, then you're good or bad. But I think that would be super interesting to investigate if there is a flipping point when you just can't rely on the resources or the network interactions, or let's call it the youngish state, right? And I also know that some TDCS studies from the Markus Mainzer group, where they could show that facilitatory brain stimulation helps to regain the more youngish-like network pattern. But this was, again, old subjects versus young. And I think it's relatively unclear what happens during this middle-aged subjects, but would be super cool to investigate.
Yeah, also, so the middle age for sure to see that trajectory and maybe get a tipping point idea.
Exactly.
But also the very young. So some studies have reported that language activity in children tends to be more bilateral than in adults. We see that for the white matter as well, that it tends to be more bilateral and then changes. So again, here, the question would be, when does that segregation actually, or is it the same to begin with? And then if it's not, when does it change, and when does it change back? What triggers those mechanisms?
And I have to say, I have no idea. So I guess for this, one would need a lifespan approach where you continuously scan and have a lot of subjects from all different age groups. And my guess would be that it highly depends on the cognitive function of interest. So I guess people like Angela Frederici would predict that it is very different for syntax, which comes in later than probably some of the semantic functions. It depends on what kind of task you use, how much word knowledge has to be used for that. That's what I like about semantic fluency or semantic tasks in general, that we see this paradoxical effect that the semantic knowledge increases with age, or at least stays stable, but the cognitive part declines. So it could be quite different for a task that has more inherent higher challenges to something that is not so much relying on these stable factors, right? So for example, for motor performance, I would expect that you don't have these two processes that go in the opposite direction, like the semantic knowledge gets higher, but the motor performance will naturally, would be my guess, just decline because you're getting at least less efficient, right? Yeah, but this would be super interesting, and I have no idea when this flipping point in the segregation of networks, if there is something like this. Maybe there is nothing like this. Maybe it's completely task specific. I have no idea. Yeah. But super cool and interesting questions. So also about the specificity or the generalizability of these effects. I don't know. Okay, should we continue? Because I think now we're coming to a probably even more interesting part, namely what happens in, or how does flexible neural network recruitment look like in network disorders? So when the default state is no longer reached, and not because of aging, but because of network disorders, and what the role of domain general networks may be like here. So I have two examples here. And the first is from recent work that Sabrina Turka in my group, who's also a postdoc led, and she's interested in dyslexia, which is a very interesting disorder. And as I could already say, that we think it's definitely a network disorders. So we looked at adults with dyslexia, so who have still persisting dyslexia, and had them in the scanner perform simple and complex reading of words and pseudo words. So simple was just 2-syllable words, complex was 4-syllable words, and real words and pseudo words without any semantic association. You can already see from the behavioral plots that people with dyslexia performed worse on almost all measures. They didn't have strong problems in accuracy with the simple words, but for all other measures, they performed worse. So it seems that this, of course, reflects the disorder. Now, how does this look like in the brain? Oops. Here you can see the contrast of control subjects with dyslexia. So all areas that you can see have more activity in people with in normal healthy controls relative to people with dyslexia. And this is just across all trials, so across word and pseudo word reading. And we compare this with rest. And I should say that we did an over task here. So they just read out the words aloud. And what is interesting here is that adults with dyslexia show underactivation, so less activity in the what we call extended reading network. That is areas of classical reading areas like the left supramarginal gyrus, the left ventral occipitotemporal cortex, but also the right VOTC and the right cerebellum. So there's massively reduced activity in people with dyslexia. This is nicely in line with the literature, but it extends classical reading areas. We also performed multivariate pattern analysis here, which are a bit more sensitive, and found that with such methods, an even more extensive hypoactivation. And after that, we were interested in the connectivity pattern. So we extracted the signal, or we used the signal from all the regions where we had less activity, and use that as seed regions for connectivity during reading with the rest of the brain. And we found that all these areas also show reduced interactions during reading with many different areas in our brain. So we show decreased connectivity with classical reading areas, but also with numerous right hemispheric regions during the task. So I would say this is a pattern of hypoconnectivity with both the reading network and with a lot of domain general areas. And what I find currently most interesting is the effects for the right cerebellum. So the right cerebellum shows just massive hypoconnectivity with almost the rest of the brain, as illustrated here, particularly for pseudo word reading. And I think this is interesting because some studies already suggested maybe the right cerebellum has a special role in dyslexia. And we see that it is underactivated, so it has less activity and also less interactions with the rest of the brain. And these changes or these differences in the connectivity are also related to worse performance. So they seem to be behaviorally relevant. And I think this is a nice example of how a relatively simple or a relatively standard language task, namely reading, requires the interaction with numerous other brain networks, numerous domain general areas. So we think that dyslexia is best characterized by underactivation and hypoconnectivity in the extended reading network, including classical reading regions, contralateral regions, and also domain general areas with a key role of the right cerebellum. So we would characterize dyslexia as a network disorder. And what we're currently doing is to probe the impact of neurostimulation on network activity, interactions, and behavior. And the idea is to use facilitatory network stimulation over a key hub in the reading network and see if we can then shift the activity patterns and if this is associated with behavioral improvements. But yeah, let's see if this works. Yeah, so I think this is an example of how aberrant network interactions can also characterize network disorders that are associated with bad performance during specific language tasks such as reading. And also here, it's not just the language network itself, but also its interactions with domain general networks that becomes relevant. And I should say that I'm very intrigued by the idea of stimulating with facilitatory non-invasive brain stimulation, probably not only language areas, but if the right cerebellum plays such a key role, then maybe this should be the target for potential intervention studies. And in the final part, I want to briefly talk about flexible neural network recruitment during brain lesions or after brain lesions, and the role of domain general networks here, because I can envision that this may be the most interesting part for many people here. So we know that stroke often strongly impairs language performance, can result in aphasia, and we know that language recovery after stroke requires changes in the dynamics of network interactions. And some studies from Dorothee Sauer's group, Richard Weiss's group, and other labs have already made the point that when it comes to the recovery, it's not only the language-specific network that is engaged and contributing to recovery, but potentially also more domain general networks, especially the cognitive control or multiple demand network. And this is work from in collaboration with Dorothee Sauer's group, where we were reanalyzing her longitudinal data set and were interested in neural reorganization in the lesioned language network. So we had access to 34 patients who were measured 3 times after stroke in the acute phase. So this was the very first days after stroke, then in the subacute phase after several months after the stroke, and in the chronic phase, which was 6 to 12 months or later after the stroke. And these patients had to go to the MR scanner and just had to listen to speech. So they listened to sentences, and we compared this passive listening with listening to reversed speech, because they were not able to perform a task. And I should say that these patients were, of course, relatively mildly affected. So they initially had aphasia, but of course they had to be fit enough to go to the scanner and be able to be included 3 times. So this is, of course, a bit of a selection bias. Now, what did we do there? From a previous fMRI study, we selected regions of interest where we were interested in changes in the task-related interactions across the time course of the recovery. And we selected perilesional language areas, so the IFG and the posterior temporal lobe, and we always selected individually those areas that were not lesioned. So these are the red ones are the language regions that were of interest. Then we put the right IFG as homologous area into our model, because there's this ongoing debate about the role of the right IFG in language recovery. And we selected 3 what we called domain general areas, so right and left dorsolateral prefrontal cortex, and this midline structure in the SMA and underlying cingulum. And we chose these regions because they showed increases in task-related activity during recovery. So we thought, now we look at the changes in their interaction profiles. And this is, again, dynamic causal modeling, where you can look at changes in task-related interactions and also make inferences about the direction of such changes. And this is a very complex study because we have 2 groups of patients, and we look at the changes longitudinally from the acute to the subacute to the chronic phase. And I just want to show you one preliminary, what I think is a very interesting result, namely the increased facilitatory effective connectivity, so the functional interaction from domain general areas to perilesional language areas in the acute phase after stroke. So we found that across both groups, we see in the early phase after stroke, so already in the first days after stroke, we have a strong increase in the drive from the domain general to the perilesional language areas. And this increase is facilitatory. This is something that the model tells you. If it's completely accurate, is something different. But the model says we have a strong increase in the facilitatory drive from the domain general to the perilesional language areas. And I found that very interesting because the previous studies had already shown that later in the subacute phase, we have a strong increase in task-related activity. And here we see that the the functional interaction already kicks in quite early. And now the question is, of course, is that just probably induced by the stroke, or is that meaningful? Is it correlated with good or bad performance? And here we have some specificity. We found that the stronger the increase in the individual connectivity in the early phase, the better the remaining language performance, and selectively in those patients who had temporoparietal lesions. So early increases in the facilitatory influence of domain general areas on the perilesional remaining language areas in the temporal cortex is associated with better language performance. We did not see such an association in patients with frontal lesions. I can already tell you that in frontal regions, we see something which includes more the right hemisphere, but I just wanted to keep it simple and focus on domain general areas here. So I found this quite interesting because it nicely extends what we already know from the literature, that later in the time course of recovery, domain general areas play a role. And we see here that they may already play a role quite early. And of course, what we need to look at now is not only the language performance, but also the language recovery score. The language performance just shows how good or bad the performance during the acute stage still is. And we can infer that those patients who have more interactions are those who perform better. But what is, of course, more interesting is how does this change? So which of the changes or the connectivity patterns that we see are associated with better recovery? But this is just the first result that I'm very excited about. And I think for the sake of time, we could skip the rest, although I didn't come to the ERC part now, but I feel that probably there are some more questions, and then I would just jump to the wrap-up part now, and then we could go to the questions, because I think this is much more interesting, and the ERC hasn't really started yet anyway, so I will just tell you something about the ideas, and maybe we can do this another time. So I will skip the way forward part and leave you with the wrap-up now. So yeah, what does this all tell us about flexible neural network recruitment during human cognition, or I should say language here, because I left the part that I have about other cognitive functions out. So I guess everyone agrees that neural networks must remain flexible to adapt to challenges. And we now have some good ideas that it's not just the contribution of the specialized language network, which helps to adapt to challenges, but also the contribution of domain general areas and networks. We see that neural network interactions change as we age, and some of the changes may reflect compensation, so are adaptive and are associated with preserved or better performance, whereas other changes may reflect a cognitive decline, and this is sometimes referred to as network dedifferentiation. And what remains to be done in the future, and what I want to contribute to, is a dissociation of these patterns. So dissect those patterns that are really compensatory from those that reflect the cognitive decline. Because I think if we know what is good and what is bad, then we can probably also use facilitatory non-invasive brain stimulation to support healthy aging. We found that increased demands, so during semantic fluency, require more domain general contributions and more interactions between these domain general networks. One idea that we're currently following is to use facilitatory neurostimulation to optimize network interactions and performance in aging brains. And one idea is that we may need other target areas than in young brains. So maybe especially in aging brains, we need to target more these domain general areas if they are more integrated. We also see that neural network adaptation is crucial for network disorders and recovery of function. We have an idea that dyslexia can be characterized as network disorder with massive underactivation or hypoactivation and hypoconnectivity in distributed networks, not only in the core reading network in the left hemisphere, but also including their interactions and contributions of right hemispheric regions, and especially the cerebellum. We see that domain general networks contribute to language recovery after stroke, which is super interesting. And especially a new finding now is that early increases in the facilitatory connectivity of domain general on language-specific regions is associated with better preserved language performance. And the next step is to show how this is associated with recovery, not only with the preserved performance, but with the changes in the recovery score from the acute to the subacute to the chronic phase. And finally, a way forward that I didn't elucidate here will be to probe the functional relevance of this compensatory adaptation. So it's nice to show that if I perturb here, I get upregulation there, we see hypoconnectivity here, but really to use network stimulation and probe if these changes that we see, if they are really adaptive and are behaviorally relevant. And we will do so in the future, or we're working on individually optimized neurostimulation. So to also address all these individual differences that potentially affect our neurostimulation protocols. And I think here we could stop. That's it. Thanks for listening. And I'm happy to take more questions if you have some left. So thank you.
Lovely. Thank you so much. It seems we have to bring you back when the ERC rolled out and do another
no pressure. Yeah.
Right. Are there questions in? Yep, there's questions. Okay. Over to you. I think Valentina was first.
Thank you so much. Really interesting talk. So my question was, what about the rehabilitation in stroke patients? So I guess that there are some protocols like neurostimulation protocols to rehabilitate these kind of patients. So yeah. I guess if facilitatory stimulation on the language network would improve the patient's performance. But going back to my previous question, so what about the other networks? Does stimulating other networks would improve the patient's performance?
Yeah, so my guess is yes. I don't know many studies on that. So at the moment, there's a great debate when to stimulate what. So the first issue is that across the time course of recovery, you may need to target different areas. So for example, Dorothee Sauer has shown that early upregulation of the homologous area in the frontal cortex seems to be associated with better language recovery. This also depends on the lesion side. Later, across the time course of recovery, this activation goes back to normal and the perilesional language area shows again increased activation. So one idea would be that in a time-specific manner, you may need to target different brain areas. So maybe start in some patients at least with early stimulation of these right hemispheric homologous areas and then later move to the perilesional areas. This is a very simplified model. It depends on the lesion size and site, potentially also on the type of impairment. And I think it's not clear yet which patients may benefit from what. Some researchers think you should always stimulate the remaining network regions of the core language network only and inhibit the right hemisphere because it's doing maladaptive plasticity. So it's reduced transcallosal inhibition after the lesion and the right hemisphere needs to be inhibited. And I think it depends on the timing after the stroke. So it may, if it is persistently activated, it may play or it may indicate that the recovery towards the perilesional language areas is not successful. So we probably need to change what we stimulate and how we stimulate it across the time course of recovery. And when it comes to the stimulation of domain general areas, I think it's an intriguing and fascinating idea. And I only know some first studies from Markus Mainzer's lab, where they targeted the motor cortex in people with aphasia, I think in the chronic phase after stroke, with the idea that the motor cortex is densely connected with the IFG when the parts of the IFG are lesioned, and they also reported better recovery. So I think this is also going into the direction, hey, what about other networks? And some groups like the Turkeltaub lab are also trying to use cerebellar stimulation. So also more a domain general network stimulation, so to say. And what I would find super interesting is to apply also facilitatory stimulation over these midline structures. So SMA and underlying anterior cingulate cortex, because they are usually intact in stroke patients. And if they are part of this multiple demand network for cognitive control, then probably stimulating them will impact language processing and also functions beyond the language network. As you said, I mean, in these patients, usually it's not just language, they're also probably impaired in their executive performance, they are slower. So maybe if you stimulate these more general network resources, then we can also improve specific function. That would be my guess. Yeah. I don't know who's next. There are so many hands up.
Yeah, so I have 2 questions. One is, I didn't get if the strength of the facilitation of domain general regions depends on also on age. So the older is the person, for example, the facilitation of that domain general networks is lower. Is it something that you studied as well?
That's a good question. So what we did with in 2 cohorts is study interactions between the semantic and domain general networks during noisy listening conditions. So to challenge the network and then have sentences with strong semantic content predictability. And there we saw something interesting, that the overall network activation and interactions are reduced in aging brains. So I think the aging brain is a relatively complex model, because on the one hand, we see these shifts, that we see more interactions between domain general networks, but in general, we see overall less activity. And it's not so clear where this comes from. It could come from general, what they call in the literature, de-differentiation. So the network is not as strongly activated anymore. We see that some of these changes can be compensated for if we, and this is a very simple thing, if we just probably adjust the hearing thresholds for the subjects. So of course, in the fluency task where you have a visual task, that does not matter. But if you have speech and noise, then for older subjects, sometimes it already helps if you increase their the perception threshold so that they can hear things more properly. And if you individually adjust this, then they perform almost as good as young subjects. What we generally see is that they are always slower in task performance, independent of their task. But coming back to your question, I think that's hard to address. We have different aspects here. We see overall, in general, less activity and less connectivity, but the activity and the connectivity that is there is more shifted towards these domain general networks. Does this answer your question?
Yeah, thank you. And the second one, I still can't figure out why there is increase in the opposite hemisphere in terms of, like, at the level of neurons and blood supply. So when you inhibit neuron with a TMS, so it means you are kind of inhibiting the neuron to consume the oxygen from the blood, right?
Yeah.
So why is the other hemisphere increases the blood supply on the other? You're kind of blocking, I don't know, too much of oxygen in the left, and then that oxygen just travels to the right hemisphere, or I don't understand that part.
Yeah, me neither. So I think the problem is that the mechanisms how TMS works at the cellular level or neuronal level are not well understood. And there are several ideas what could happen. One idea is that if we stimulate with a repetitive protocol for a longer time period and separate the stimulation from the measurement afterwards. So we first stimulate the area and then put the subjects in the scanner. And this is all the experiments that I showed follow this way. Then we induce short-term plasticity, and that should follow the logic of Hebbian plasticity. So what we induce is synaptic plasticity, which means that it's just a shift in the balance of the network. You inhibit one area for a continuous time, and the brain adapts to this by showing more activity in the other area. So what some people think this is an adaptive synaptic plasticity, what we are observing here. Other people think this homologous changes rather reflect the reduced transcallosal inhibition after the perturbation. So if you stimulate the left IFG and inhibit it, this will reduce the transcallosal inhibition from the left to the right, so from the dominant to the non-dominant hemisphere. And then what you see reflected as increase in the bold signal is just that the right hemisphere is resolved or freed from the normal inhibitory influence of the dominant hemisphere. And it's not so clear. I would not think that our results reflect the latter explanation. I think this is unlikely because then it would not be associated with preserved or better behavior. And it does not explain why the right hemisphere in turn takes an active facilitatory influence on the left hemisphere, what we showed in some of the studies. But you're right, the mechanisms how TMF work on the neuronal level and why the patterns are like this are not clear. And I would think of them more as a shift in the relative contribution. So all these areas are somehow active, some are above threshold, some are below threshold. And if you perturb one area, then you don't silence the neurons, you just shift the relative contribution of this area. So this goes down, which can also be reflected in deactivation in your bold signal. And then the other areas just gets more activity because it's more needed. So it's part of a network or of a region that interacts with the other area. And it has more activation because it gets more relevant for the task. This is, but at the neuronal level, I can't say how this is because this is not known yet. Yeah.
Yeah, perfect. Yeah, thank you so much, really.
If I can quickly intersect, sorry, Ma. Just to follow up on that question, or rather the explanation of it. So you said that everything is kind of connected and a network will affect the entire network. And that reminds me of the holistic model of the brain. So you have the localizationism, associationism, holism.
Yeah.
And am I correct in assuming that your assumption is that the brain kind of functions in a holistic way and everything is connected? And if you have a lesion to one part, or if you inhibit one part, it has a knock-on effect on all the other.
I'm not sure if I would go that far. So I would say it will certainly affect not only the specialized network, but always also its interactions with other networks. But if it affects the whole brain as a holistic concept, then if one thinks in extremes, then one could say, yeah, but then you don't have networks anymore. That I don't believe, because we see that we have these relatively specialized language network. But I think language is a good also model to study, because whenever you perform a language task, you're not only performing the language task, but you need to have strong attentional components, working memory, cognitive control to suppress other incoming stimuli. So I think language as a higher level cognitive function naturally uses many resources of distributed brain networks. I'm not sure if this holds for a very simple motor task, but even there, I'm not a motor expert, but I would think that it's already fascinating that if you tap your finger, then you don't only see the strong activation of the primary motor cortex, but also of surrounding motor regions. So already there you have a network. And of course, if you're required to tap your finger in a specific rhythm, then I would also assume that requires attentional resources, right? So in a way, yeah, I think I would take a more holistic perspective, but I still believe somehow in brain networks, and that the core language network, I mean, we wouldn't see loss of function if everything would be completely redundant, right? So if the brain was just redundant, then a stroke would probably not affect any specific function, but just the overall performance efficiency or something like this. Yeah, but it's an interesting debate, and I'm not sure where I stand in this debate.
It took me a while to figure out where the changer is and where I fall on the spectrum.
Yeah.
Hello, thank you for the talk. It was really interesting.
Thank you.
I was thinking that the network adaptation that is observed in aging, it's probably more long-lasting, more permanent, while the network adaptation after TMS or after brain stimulation, it's more transitory, more
yes,
fast.
Absolutely. Yeah.
So I was thinking how this could impact in behavior, like how these two types of network adaptation can have an impact in the behavior.
Yeah, that's a very good question. So the all these effects of non-invasive brain stimulation that I was referring to today are only the direct after effects of a single non-invasive brain stimulation protocol. So I would think that also at the behavioral level, sometimes we don't see anything at all. And if we see something, then the effects are also very short lasting. So I think if you want to induce more permanent changes that are also potentially therapeutically relevant, then you definitely need to stimulate repetitively. So you need a lot of sessions spaced across different times. This is something we know from depression, where TMS is already used as treatment, and where they really use high intensities and high doses of non-invasive brain stimulation repeatedly, sometimes even 2 protocols on the same day over several weeks. And then you can also induce behavioral or changes in these mood rating scales that outlast the stimulation for several weeks or months. But if you only have a single protocol, then the effect is very short lasting. So if you really want to mimic what's going on a more longer time scale, then of course you need to stimulate repeatedly. But I think it's nevertheless interesting to see if we can build good models with these short-term plasticity, with single sessions of brain stimulation that we can use to inform models of more longer scale changes in compensation, and potentially also inform ourselves where to stimulate. So which hubs would be good candidate areas for facilitatory neurostimulation. Yeah, but it's true. So these on a time scale, they are very different and likely not comparable. Also, sometimes when I read that some people say, oh yeah, we use a virtual lesion to mimic a stroke, I think this is wrong. This is just a wrong concept. We are just perturbing an area. This is a very transient effect. It doesn't mean that you have a complete loss of function. So with TMS, you can't mimic aphasia or something, or a brain lesion. Yeah. Now, this is something that I'm very interested in at the moment, also to not only look at these direct consequence of a stroke or then the changes across the time course of recovery, but also more long-term plasticity induced by brain lesions such as tumors, which have slow growth. Yeah. I think we have another question by Sabrina. Hello.
Hello. Yes, thank you. Really interesting in your talk. So, in your study on persons with aphasia after stroke, do you control somehow also the linguistic rehabilitation of these persons? That I think they do.
Yeah, so these patients, this was the spontaneous recovery. So, but of course they will, when they are in the late subacute to early chronic phase, they will also go to a rehab hospital. So, and it would be unethical to prevent them from undergoing therapy. But so what we didn't study with this cohort is the effects of language therapy itself. So we didn't specifically look at the therapy-induced changes, but focused more on the spontaneous recovery. But this, of course, doesn't mean that none of the patients had access to therapy, because that would not be possible. Yeah. So we have to take this into account. And I can say that here for the acute phase, we didn't take this into account, but for the later recovery score, so we measure the changes in our aphasia battery, so how their task performance changes. And this will, of course, be impacted by the amount of language therapy that they may get. So those patients who recover better or have good performance increases, recovery scores, are likely those that also respond well to therapy and probably had access to treatment. Yeah.
Thank you.
I had one small question left, if it's more one of those way forward questions anyway, so maybe to round it off, because the picture looked so clear and so informative about dyslexia. I was just wondering if you're planning in future studies to do the same kind of analysis for other disorders, since, yeah, SLI, whatever it might be, any cognitive disorder, to help understand what is downregulated or what part of the network is under firing, etc.
Yeah, that's a good question. I was surprised myself how clear the picture looks like and how strong these effects are. So if we look at the individual subject data, then we see the same pattern almost in all our subjects. So it's really, I wish the non-invasive brain stimulation results would have the same strength, but yeah, usually that's not the case. No, I relatively recently started with these with this dyslexia studies, and I think we want to go more in this direction and also combine, use more brain stimulation to better understand what's going on and where we may find angles to help. But so far, the models that we investigate are dyslexia, stroke, and brain tumors. And this is already a lot, but yeah, it would definitely be fascinating also to look at other disorders and maybe identify common patterns across different disorders, right? So especially the learning disorders. So maybe they have a common core, but I have to say, I don't know this, if this is the case.
Yeah, or even within stroke, all the different sub parts, which of course is again another box of Pandora, how you classify the different kinds of aphasia. But I think the community would be highly appreciative.
That would be super cool, but I have to say that getting access to these stroke patients is already, I mean, doing patient work is so much more challenging than just inviting nice university students who need a bit of money, right? And participate in all the research. So I think working with stroke patients, you have so much more variability, such strong exclusion criteria. So it's really hard to find a large cohort of patients. So I have to say, I'm really glad that I'm collaborating with Dorothee Sauer, who's doing great jobs, but otherwise I would not be able to investigate any patient at all, because it's really hard to get access to them. Yeah.
Any final questions? No. Okay. I have a long list for you, but I'm gonna
and also approach me afterwards, of course.
Great. Thank you so much. We're well over time, more than academic quota. So I do apologize for everyone. We had about 10 people on YouTube as well who watched us. So thank you very much for staying with us, even though we ran over time. Thank you so much for joining us this afternoon and presenting your work. And we look forward to your next talk on the ERC project soon.
Thank you. And sorry for running out of time, but I think the discussion was really worth it. So I really enjoyed it.
That's fantastic. I never try to kill the discussion until we get a bit too far in time, and usually it carries on after. But I just want to give everyone who needs to go and pick up kids or do whatever, give the opportunity to leave. So yeah, thank you everyone. And if you